All right, so yeah, my name is Alex Mietke. I have just joined very recently the theory physics here in Oxford, and I'm really enjoying this a lot. And I want to tell you a little bit about the research that I have been doing, that I am doing, and also introduce this presentation a little bit with some general thoughts on what is going on in the field, and making some arguments why I also think that physics has maybe some interesting things to say about processes like these. So what you see here is, probably really one of the atomistic unit of developing an organism. What you have to do, it's a single round cell that we begin with. We are looking at a cross section, and of course biologically what is happening in this cell division is that the genome that you see also slightly in some different contrast here in the middle, through this process, gets distributed to the two daughter cells that this movie ends up with. And so this is clearly one of the many biological functions of this process. But I hope you can also agree with me that it is also a very fascinating physical process. If we think about this as a material, it seems to be a material that is able autonomously to change its shape in a very coordinated way. And our experience with changing the shape of a material would maybe be something like this, right? If we take a rubber ball and I asked you to change its shape, what you naturally would probably do is to squeeze it, and that's would do the job. But what that means is you have to apply a force, and obviously there's no one squeezing this cell that you just saw in this movie. So somehow this cell is able intrinsically to generate these kind of forces that are necessary. And clearly what must happen is that somehow all the molecules that are interacting in this system must be able to produce these forces that are doing this. And when I talk about interacting molecules, we're very quickly in the realm of chemistry. And indeed, as we had also heard before now in several forms, there are processes that convert through chemical reactions some fuel into forces and expel some of chemical waste. And I will specify this a bit more moving on through this talk. And so what is it that we have to think about when thinking about the physics of these biological systems? Julia had already mentioned some very important thinking that Schrödinger has done now precisely 80 years ago, laying out this basic idea that obviously what makes living systems different from that system is the fact, physically speaking, they are kept away from thermodynamic equilibrium. And I want to just add here a little bit more and connect this maybe also with your undergraduate thermodynamics memories. If we want to keep a system out of equilibrium, one way we were taught this can be done is through what is called a heat engine. And at the core of every heat engine is a temperature gradient. So we have something hot, we have something cold, we connect this to the system that we want to study, and because we have this ongoing hot and cold, we can sort of do work with our system, okay? Now, the interesting thing in a biological system is that somehow everything is packed together, right? Because there is no, no external temperature gradient that is applied in most cases. And in fact, it is usually not temperature that maintains these fluxes from hot to cold to extract work, but there are chemical reactions going on that in principle generate the same picture. And there is an analog to temperature gradient. This is called the chemical potential, but all what that means is you have to put energy into the system in a cell that's nutrients, for example, that this particular cell you saw got from its mother. And this allows it to some molecular processes, use these chemical potentials, turn it into work, turn it into forces. Okay? And this leads to all this fascinating, beautiful kind of self-organization that, Julia and Adrian had talked about. Now, how is this molecularly implemented? I'm also here, not necessarily have to go in all detail because Adrian had mentioned this a few times and you as well. So if we look a bit closer at the surface of the cell, what we will see in the most simplified physics picture is a polymer network where I sort of sketch these actin, these sticks here. They are called actin filaments, as we heard before. And on these filaments, we have these very specific proteins called motor proteins that exactly do this fuel to waste conversion by changing the conformation of some molecules that are floating around. These conformation change turns them from ATP into ATP. But the point is, if you have such molecules walking on these filaments and they pull on one end and on the other, they are sort of naturally able to inject forces into this material. And you can also imagine that when I have a single cell doing this, and I have now a tissue where many of these cells are connected, that these forces can permeate through the tissue and generate motion also on a larger scale. And then you might find other mechanisms by which the strength of these forces can be controlled. And so what does this now look in practice? I add to the list of, I hope you found beautiful movies that you had seen today. It's an organism that we had seen before already. So this is a zebrafish, very popular model organism. And what you see here is the same process, but it happens on the surface of a sphere. So we're looking on the left side from here. The movie on the right looks at it just from the other side, but it's the same process. Each white dot here is a cell. Okay, you can think of this as a cell. And obviously, each of these cells now is undergoing this kind of cell division that you saw in the first movie, just a single cell doing. And in the same time, they are moving around and coordinating their position in space and time. So it's a beautiful process. And just another example to add to the list. Again, remember, we have a well-defined sort of molecular system in terms of what produces forces that sits, in this case, on the surface of an ellipsoid. Again, each dot here is a cell. And when we play this movie, we are looking at the development of what is a flower beetle. And it has a kind of other, also an interesting geometry, but it's different than what we had seen before. Actually, it makes a little compartment here. And later, the embryo develops inside this compartment. So it makes sort of its own cavity to have to snuggle in and develop. So I just want to make the point, right? So there are a lot of ways how these forces can be translated into some very interesting morphogenetic processes. And so the question that I want to specify a little bit to what was asked before is then, what can now physics actually contribute here to understand the development of living systems? And I would alternatively title this as maybe what's the condensed matter conundrum of biology, and illustrate this with another modern organism yet. This is C. elegans, single cell embryo on the left, fully developed organism here on the right. It's a sort of millimeter long worm, half a millimeter long worm. And it has, as an adult, about 3,000 cells. And what's interesting to note about this, we know almost every microscopic detail of this organism. We know precisely its genome. Of course, from humans, we also know the genome nowadays, but this was one of the first organisms where this was actually fully decoded. But we know much more than this. We know actually of all these 3,000 cells that emerge in the adult organism, exactly how they got there. It's very reproducible. We can follow and predict the position of all of these 3,000 and a few cells. On top of that, we know precisely its neuronal network. So it seems like we have all the microscopic information about this organism available from single cell to final grown adult. And still, we have no close picture here that would bring us from the single cell to such a picture as an emergent self-organized process as we would think of this when we look at it, okay? And I'm calling this the conundrum, the condensed matter physics conundrum in a sense because there had been a very influential essay 50 plus years ago by Phil Anderson, who was a condensed matter, dead condensed matter physics, non-equilibrium condensed matter physicist. And he made this point in the context back then that some people were proposing that what needs to be done is to understand the fundamental interactions of nature, say particle physics, as good as possible, and then we know everything. Okay. And he was making the point that this is not exactly true, but because as soon as you have many interacting units, you have something called emergence and collective organization that comes out of this. And you may actually have to think in different terms about describing these collective phenomena and are not able to necessarily derive them from the microscopic laws of your system. And that's where physics, of course, can really help biology, because biology has the same issue as I tried to explain or convince you of with this organism on the top. And so what are the research questions then that I'm looking into? So very bluntly, so I want to understand how a cell divides, how a tissue folds, and of course when I say how do they do this, from my perspective, what are the physical principles that facilitate these processes, and then taking this together, how this can help an organism to develop as a whole. And certainly I'm also a theoretical, I am a full-time theoretical physicist. So what I really want to understand is what I can learn about these things using theories of active matter. And then maybe more excitingly, and Julia also made this point quite nicely, using biological system to inspire our thinking about what theories of active materials have to actually look like and what they have to be able to describe, okay? And so I just sketch essentially this last box here on the slide, because it's really crucial, as you hopefully could gather already up to here, that this work is resulting always from very close interactions between experimentalists and theorists that have very mutually fruitful interactions, or when these interactions are mutually fruitful, this is mostly where the most exciting and interesting things happen. And so this was kind of a general introduction, so I want to now turn a bit more to what I put into my title and want to tell you what is chirality. And maybe many of you have already heard about this. For the purpose of this talk, what you have to think about it is, think about an object that when you mirror it, it's just not the same object anymore. And this can be very nicely done, of course, with hands. We mirror a hand. We have another hand that is this mirror image, and clearly they can't fit on each other if you sort of keep the orientation in the same way. And another way to say that is we have in this case a broken left-right symmetry, because left and right in our body, and in fact in most organisms, is not the same, okay? And you can already ask yourself this question: if we start as a round cell, as an embryo that doesn't know a direction, no front, no back, no up, no down, no right, left, this must be somehow implemented, because we end up all in a well-defined, with a well-defined morphology. How is this symmetry breaking actually controlled and implemented in biology? And that's why chirality is in general a very interesting field to study in the context of active matter. There are many more examples. I would really say that the living world as a whole is chiral. As a few examples, you can look at the patterns on shells and snails. There are like spirals. If you think about a spiral, if you mirror it, it won't be the same spiral. You see spiral patterns in plants. Also more subtle, so this is a crossbill. This is a bird that has evolved its beak to grow a bit more to the left on the top and a bit more to the right on the bottom, and it gives this bird an edge over catching little bugs out of narrow spaces from trees. Something I learned preparing this presentation. And so if we want to link this a bit more to the research I've been doing, we have to get a tiny bit more technical where the chirality is in the system that is interesting for physics. And one aspect is the actin. We had heard now several times about the actin. Again, this all these molecules in the surface of the cell. If you look at them a bit closer, these molecules are built from monomers, but they're not built as a straight stick, but there's a little pitch whenever a new monomer is added. And so you end up with something that's more like a helix, okay? And the helix, just like a spiral, has sort of this notion of handedness and chirality to it. So you have molecularly inbuilt into this cortex chirality. Okay? And then something I will get to later, a little bit more detail. This is the force generating structure we had heard a lot about. This we haven't talked about, I believe, so far. Are cilia. So I'm talking here about the sperm cell that must be propelled somehow through a fluid. So it has a little appendage, and there is a molecular motor machinery in this appendage that is very similar to the one that you see in the cell. The net outcome is that you have a machinery that produces forces that it exerts then in this case usually on surrounding fluids and either propels this cell or if the cilia are stuck somewhere, moves fluid around. But I will show you in a few minutes, a bit more details about this case. So let's first look at what this chirality here can do for a cell. So remember, what we are now looking at is again an embryo of C. elegans. I just show you another image where really these filaments are very clearly labeled in a specific way. You see this nice anisotropy, this sort of filamentous structure. So each of these white lines are essentially these purple actin filaments. And when now these myosin motors are walking across these filaments, you get a very sort of complicated dynamics because now these myosin motors, they will pull and push on actin, move the cortex around, and this gives you some sort of merge dynamics. So this is still a single cell, and we're looking sort of on the side of the cortex. So this is a single cell. Now this single cell has divided. So we have now one cell here, and we have one cell here. This is after the first division. And now something interesting happens. So it doesn't just divide anymore in splits, like everything we had seen so far. But if you look carefully when this movie starts playing, it will divide along this axis. Okay, this is not too spectacular, but it's actually also having motion in the top cell that goes rather to the right, and motion in the lower cell that goes rather to the left. Okay, let's play this movie. Right, so you have very significant motion that again has sort of this notion of helicity, of handedness, of chirality, and the indication is here that somehow this chirality of the actin that I described gets translated here through these active processes into a motion, specific type of motion of the cell. And what you hopefully also notice is that the division axis, as it divides, is rotating. So I start as a cell that is essentially parallel to the left boundary of this video. At the end, it's somewhere lying diagonal in this video. Okay? And this is quite remarkable because at the beginning of the movie, you have two cells that sit on a line. That means all that this embryo at this time actually knows is what its front and its back will be. But it doesn't know anything else. Any other direction looks the same up to this point. Okay? But the moment you have this kind of division and this sort of rotation, you're selecting a specific plane and you actually finally broke morphologically left, right, up, down symmetry as well of the organism. So there's a very clear first moment where suddenly this organism starts to have not just the front and the back, but actually also a front, left, right, up, down. And so this is the point I just wanted to make. And we looked a little bit more in detail, actually, what is happening here. What I want you to take away is there are cells that divide without rotating. Okay, so there are specific ways that we characterize this, or the experimentalists characterize this. And there are cells that divide without tilting their axis. And the point is that this tilting of axis is very well correlated with having these counter-rotating flows that you see on the surface of the cell. So this is what this plot here is showing. So more counter-rotating flow basically goes to the left. And you see the more of this we have, also the more we are rotating the cell. So there's a very clear correlation between flows and rotations. And the physics here, the physics question is, what are the torques essentially in this problem that translate these flows actually into the rotation of the cell? And I spare you the details, but I tell you that what we arrived at after evaluating carefully how the torques balance in such a process, what we call the bulldozer model of cell representations. And I'll briefly explain why. So we arrived at a formula that says the amount of flow of this counter rotation is proportional to the rotation of the division axis. Okay, this is essentially what you saw in this plot. Yeah, the more flow I have, the more rotation. So this comes out of the theory. But the physics is in this prefactor here. And this prefactor says, what I have to look at is the friction on the two sides that this dividing cell is in contact with the egg shell, because it's particularly in contact with the egg cell on two opposing sides. And whenever I have a difference between these frictions, this number will be non-zero, and I will arrive at something that will rotate in space its axis. And why do we call this a bulldozer model? Because a bulldozer that you want to rotate on the spot functions exactly the same way. Okay? So now the flows of the cell are represented by the motion of this chain. Okay? What I call here the two sides of the shell is basically the top of the chain and the bottom of the chain. Okay? And if a bulldozer stands on a street, essentially the friction is infinite with the street, and there's no friction on top because just nothing on top. And you see in this case, in this limit, this just becomes 1, and I'm perfectly translating the motion of this chain into a rotation of this bulldozer on the spot. And it's this same principle that this cell now exploits to convert molecular chirality of the actin into chiral cell flows into a rotation of the cell axis, which ultimately, for the first time, breaks morphologically the left-right symmetry of this organism. And so now I move to the sort of second example of my research. I had mentioned there is this other very interesting force-generating structure in biology called the cilium. The types of molecules and motor protein involved are different, but they are the same principle. And where's the chirality here? Well, if you take a cross-section through this cilium, what you will find is molecules arranged again in a kind of helical structural way. So this arrangement here is called the axonin. This goes through the whole flagella, and it also has a pitch throughout. So there's an inbuilt chirality as well into these cilia. And I'm not interested so much in a single cilium, but actually what you have a lot in biology are what are called cilia carpets. So those are structures that have different kinds of functions, but they are structures where you have many of these beating cilia together. So they are sort of very densely packed and form these kind of carpets. So what we have now is a surface with a lot of cilia that beat, and keep in mind now this beating will have some chiral nature to it. And in what context can something like this be relevant, or why at all we think this is important to understand how these carpets are affecting flow and vice versa. So there is a very interesting condition called situs inversus. For anyone of you who had not heard this before, so about 1 in 10 to 20,000 people among us have a completely mirrored internal body plan. So essentially all organs, everything is perfect. It's not just on another side. The whole morphology of everything that's internal is mirrored. And many of these people live a healthy life and don't even notice. But what has been found is that this is often related to a specific gene mutation in human that codes for making cilia, okay? And so the idea is here that at some point in development, these cilia will be used to generate some kind of flow that, patterns the tissue. And if this flow goes in the wrong direction, downstream, when you arrive at the newborn, you have produced a human being that has a completely mirrored internal body plan. Okay. So we go a little bit more lo-fi and look at the starfish embryos. We had seen those already. Because they also have cilia carpets, but a slightly different purpose. So this is a microscopic image where you see each of these little, what looks like threads that come out. They're all cilia, and they just keep beating on the surface of these little embryos. And what this does, it allows these embryos to swim. Okay. Of course, this doesn't look anything like a starfish. This takes quite some time to get there. It's really, we are just looking at this early stage where it's somewhat a benign ellipsoid. But the point is it can swim. And what you maybe can see already in this movie is it's not just swimming, it's also rotating. So it has a forward motion, but also a sort of corkscrew aspect to it. And what we got interested in here is that these embryos not just swim, but they also quite often approach the surface of the fluid, between the fluid and the air, and then they don't really swim, but they just keep rotating. So that they maintain the rotary aspect of the motion, but they don't really swim away anymore, okay? And what was particularly cool is now if you put a lot of these embryos together, a lot of them come to the surface and they make these amazing, what we termed living chiral crystals. We call them chiral crystals because this rotation direction here of this embryo is always the same. So you don't have some spinning right, some left. It's always the same. And of course, the suspicion why it's always the same is, again, because these cilia have a hard-coded handedness from the molecules they're built from. And accordingly, this rotation is also hard-coded by the way, due to the fashion they make themselves swim. And the idea was of this project not so much necessarily to understand what's the biological meaning of this crystal formation, but really thought this is a great opportunity to do active matter physics, because we have very good access to understand these embryos, how they are moving in the fluid. And so the question is now, can we understand from their properties what the properties of these emergent crystals would be, and if they are different from crystals that we are used to? Okay. And one way to do this, of course, they are in the fluid, so we want to understand how the fluid around them moves. What you see here, hopefully in this movie, is there are little particles around, so it's a bit, there's some structure. These are little beads that are also put into the fluid, and the assumption is now that these beads move like the fluid does. So that means if you track the velocity of these beads, you know how the fluid moves in the surrounding. So this is called particle image velocimetry. And if you use that, you can get a map like this that tells you regions where the flow is very fast and regions where the flow gets slower. And so naturally, it's fast close to the embryo, it gets slower moving away. But what's more important is that it's now giving you immediately an idea why at all there would be a formation of these crystals, because this fluid, what you see is it's drawn into the embryo from all sides. Now imagine you have another embryo sitting here. Naturally, it will sort of be drawn to this one embryo as well, and all of these embryos are doing it. And so naturally, you have a mechanism to nucleate these kind of crystals, and there are actually also other biological systems, Volvox, a specific algae, that has a similar flow phenomenology that was studied in Cambridge in quite some detail some years ago. So this is a single embryo. When you want to understand crystal formation and the properties of a crystal, you need to understand a bit about how these embryos are interacting. And the simplest interaction that you can have is in a pair, okay? And essentially one important thing now is happening. It's not just that these embryos independently spin, but the moment they come close to each other, they start dancing around each other. So they're orbiting around each other. And qualitatively, the reason is that between them, there is still some fluid, but because they're so much nearby and both want to rotate, the kind of torques and forces that emerge hydrodynamically make them walk around each other. And studying this problem quantitatively in more detail, you can come up with a theoretical model of this process that captures in many ways the kind of crystal formation we see in experiments. So now imagine this is the top view. We have disks. They are following the interaction rules that we have determined from the kind of experiments that I just shown you. And the color code here is the rotation frequency of these embryos. And one thing maybe to note, you see that they slow down quite a lot. Yeah? So they start at a rotation speed that you see by eye. Here, a single rotation of an embryo in the final crystal takes about 3 minutes. So you don't even see anymore that they rotate, but they still do rotate. And there's some nice observations that you can check that sort of to test your intuition in a way. So what I show you here is the final state. The color code, again, is the amount of rotation, the speed of rotation. One thing we immediately see that the boundary embryos are usually faster. That's not so surprising. Okay, so those are embryos that have less neighbors. So in a sense, they have a less hard time to rotate because there's less to work against. And this is something you will find also in the experiments. So you see these boundary embryos rotating much faster. You don't really see their rotation. Those ones are much slower. But then interestingly, there are also some fast ones in the middle. So you see some very bright ones here. If we look in the theory, what we find is that those are ones that are rather on the smaller side. also makes sense. They have sort of less contact with the surroundings, so it's easier for them to keep rotating. And that's also something you see in experiments. So here's one mark that is a bit more small, a bit smaller, and you see it's going quite wild. Whereas the other ones, again, in the surrounding do their usual rather relaxing spinning. And then finally, we have the opposite thing as well. We have some here that actually rotate in the opposite direction, okay? And if you look carefully, what is happening is you have some of them that are they're large, statistically speaking large, and at the same time, they don't want to spin themselves too much. Now imagine this embryo in the middle doesn't want to spin at all, and all that happens is that the neighboring ones are at full speed spinning, okay? If you look at the interface, what they will all do, they will spin the inner one in the opposite direction that it's usually turning. And this is something we see in experiments as well. So you have two examples of two big embryos, and you see that they spin precisely in the opposite direction of what all other embryos doing. And so this gives us a little bit of a sense that we are capturing most of the physics here in the right way. And so finally, what was a bit the kicker of this project, this is sort of very microscopic how I describe the system now. But we were wondering, what are sort of the macroscopic properties of this crystal, okay? So we know that a lot, condensed matter, solids, arrange often in crystals. We can think about their emerging material properties. Can we say something similar about these kind of crystals? And when we looked at our theory in more detail, we found quite a surprise. So back in the continuum mechanics courses that we take, we are usually told that for such an isotropic solid, all you need is two material parameters to describe all its properties, okay? And Adrian has mentioned this, and that is usually true. But what we found is that our crystal somehow requires 4 parameters to describe in full all its elastic properties. So we are very carefully going back to our Introduction to Solids courses inspired by my daughter. And we found actually it's not a problem at all. The reason that this is allowed here is that what one is told about these 2 parameters also assumes that the system is at equilibrium, okay? And our crystal is not at equilibrium. We have living organisms that are pumping constantly energy into the system. And as a result of this, of chirality and the non-equilibrium nature, we are allowed to have these additional material parameters. And actually, they are referred to as odd elastic parameters. And this was a theoretical concept that not so long ago was worked out by a group in Chicago. We did some beautiful physics that we could kind of find again in these living crystals. And then to just finalize and end with 2 slides of some exciting movies and dynamics that these crystals also take, all the consequence of its complex properties, they spontaneously oscillate. So this is this full crystal, and I hope you can see that there are some sort of large-scale displacement waves that are traveling through this crystal that we observed. And normally, again, in a normal conventional crystal, this would not happen because this is an overdamped system. It's a very small length scale, but here the activity injection, this is sort of a data processed version of this, allows these displacement waves to spontaneously pop up in such an overdamped system, being a consequence of its non-equilibrium nature and being a consequence of its chirality. And with that, I come to the conclusion. I hope I could convince you at the beginning that active matter theories are a really powerful tool, right, to study complex phenomena in biology. That I showed you that chiral cortex flows translate through friction into some morphological left-right symmetry breaking. And finally, I told you about these new living crystals that we found that have some unconventional elastic properties. And I really want to thank specifically the people that were involved in this project that I talked about, excellent collaborations with Lokesh, Zehan, and the faculty group at MIT, and the Grill Jülicher Lab, and Jörn Dunkel, my supervisor at MIT. But then also I want to thank the Rudolf Peyer Center here in Oxford for being absolutely amazing colleagues. and I'm happy to take any questions. I don't have postdocs to take the nasty ones, so please. Thanks. So.